Mas malinaw tayo ngayon. I made sure. Na- Speaker. Hello?
Wow, sana may flower.
Hello, mic test? Mic test? Naririnig po ba ako sa online world?
Yes po, sir.
Ilan siya? Yan, bagas na.
Ano raw masasabi mo sa iyong mga classmates?
Thank you, everyone.
Okay, so good morning, everyone. So to those in the online world, can I see a thumbs up if you can hear me? Ayan, naririnig po ba ako sa online world? Hello,
Po,
hello?
rinig po.
Yes, all right. Okay. Ayan, so I will now be using the microphone. Okay, so I hope those in the online world can also hear me well. Okay? Ayan. So, dahil may birthday, magki-quiz talaga tayo. No? Now na. Okay. Um, hindi, joke lang. Ayan, but we will do have a quiz probably after the break, the morning break. Okay? Yeah, kasi ang plan ko is we finish all the structures in the morning and then we'll have the metabolism in the afternoon. Hopefully, we get to do that. Okay? Ayan. So, ay, sige po, sige po. Ayan, sige sir. Ayun, yun. Yun. Okay. Ayan. So, ayan, last night, I actually went outside. Pumunta ako sa SM. Salamat sa recommendations. No? So, I went to downtown. Ayan. So, actually, nag-mall hopping ako kahapon, no? And I searched for the adapter. Yes, gumagana na ang TV. Ayan. So, ayan, no? Na-realize ko kasi na something was faulty yesterday. And I know I can deliver it better if you can see. Oh, okay lang yan. Uy, may ano na, may dent na yung ano, yung aqua flask. Ayan. Okay, ayan. So I know that you will understand it better, no, if you can see clearly the pictures, 'di ba? Ayan. So effort. Ayan. Pero, of course, para pantay. Yan yung nakikita ng nasa online at saka nasa physical world. Alright? So, yesterday, we already finished tackling the structure of your um, nucleotides, and nucleic acids. Okay? Tapos, we already started with the central dogma. Tapos, we are already done with replication. Now, I will be interjecting some items which are related to biotechnology because biotechnology principles are also included in the board exam. No? Ayan. So I'm just trying to prepare you for whatever is to come. Okay, so let's begin with the PCR. No? So PCR is polymerase chain reaction. So basically, PCR is an in vitro process which resembles the replication process that occurs naturally in organisms. Okay? And so it is our way to magnify, no, or to amplify the amount of genes, no, that we have already identified from your organism. Okay? So ito yung pinaka workhorse na tinatawag, no, of biotechnology. Ayan. So actually my background is in biotechnology. So I really like to discuss this one. Pero medyo briefly na dadaanan lang natin. Okay? Ayan. So again, no, this is the workhorse of biotechnology because it amplifies, no, genes that have already been identified. Okay, so it is an in vitro process. Okay, so take note of those terms. No, in vitro amplification of DNA. No, that is your polymerase chain reaction. Okay, now polymerase chain reaction is similar to the process of replication. Okay, and it uses a thermocycler or a thermal cycler. Okay, now remember that there are steps, no, in replication. Okay, so what are those steps in replication? You have your denaturation. Okay, denaturation is initiated, no? Denaturation is initiated by your enzyme, which is DNA helicase, no? Ayan. Together, of course, with DNA gyrase, no? Ayan, tandaan niyo yun. Remember that DNA helicase is a part of your pre-priming complex. Okay, yeah. So in the pre-priming complex, you have there the RNase primer. Okay, you have the single-stranded DNA binding protein and the DNA helicase. Okay, yung DNA helicase, its purpose is to break the hydrogen bonds between the bases in your duplex DNA. Okay, tapos habang nangyayari yon, no, your DNA gyrase, also known as your DNA topoisomerase, okay, also relieves the supercoiling. Diba? Sabi ko kahapon, pag supercoiled ang DNA, mahirap siyang ma-replicate. Okay, yan. So mahirap din siyang ma-transcribe. Okay? Kaya ang tawag sa kanila ay mga heterochromatins. Okay? Ayan. So you have there the DNA gyrase and the DNA helicase, which helps, no, in the unwinding of the DNA. That's the first step. That's the initiation or the denaturation step. The next step would be the addition of the RNA primer. Okay, so the RNA primer attaches or anneals onto the template DNA. Okay, so di ba dumidikit yung RNA primer doon sa template DNA. Okay, and then the third step will be the elongation. No? Pinapahaba na yung RNA primer. Okay, so nucleotides are being added to the RNA primer. Okay, by the DNA polymerase 3. Okay, so remember those three steps. Those three steps are also the three steps that can be found here in they are also the three steps that are in your PCR. No? First step is denaturation. Okay. Now, denaturation would require a high temperature of about 95 degrees Celsius. Okay. Its purpose is to separate the duplex DNA into single strands. Okay. Yan. Now, the second step is annealing. Okay. So annealing is the binding of the primer. Okay. So you have a primer specific to a gene. Okay. So a primer specific to the gene binds onto the complementary site in the DNA. Okay, so yun yung annealing step. Now, the temperature varies from about 50 degrees Celsius to 65 degrees Celsius, but this one depends on the primer. Okay, it depends on the sequence of the primer. Okay, yeah, so the primer is usually an oligonucleotide, okay, which is between 80 to 25 bases long. Okay, so ganoon siya usually kahaba, no? 18 to 25 bases long. It can be longer, but it's quite difficult na, no, to make it efficient. Okay, and then after annealing, you have the extension or the elongation. So in PCR, you have a thermostable DNA polymerase, no? Dapat thermostable yung DNA polymerase, no? Why do you have to have a thermostable DNA polymerase? DNA polymerase is a protein. Okay, and we know proteins are denatured by high temperature. Okay, so dapat yung DNA polymerase hindi siya ma-denature, no? Kapag umulit yung cycle. Okay, kasi remember these are the three steps in one cycle. There can be about 20 to even 30 cycles. Ulit-ulit. Okay? Ayan. So, kapag umulit na yung cycle natin papunta sa denaturation, dapat hindi masira yung DNA polymerase. Okay? So, you place everything in one mixture. Your DNA polymerase must be able to withstand the high temperature during denaturation. Alright? Ayan. So, yun lang yung dahilan why we have to have a thermostable polymerase. No? And that is your TAC polymerase. So, may kwento yan si TAC. So, TAC polymerase, marami pinipindot. Teka na. TAC polymerase is, ayaw magpakita eh. Ayun. TAC polymerase comes from Thermus aquaticus. Okay, so it comes from Thermus aquaticus, no? Ayan, so this is actually a jellyfish. Okay, ayan, so nagkaroon siyang thermostable DNA polymerase. Okay, ayan, so Thermus aquaticus. Kaya tinatawag na TAC, no? Yung ating polymerase kasi galing sa kanya. Alright, ayan. Now, the temperature for the extension or elongation depends on the polymerase. Okay? It depends on the optimal temperature for the activity of your polymerase. Now, it can be between 75 to 80 degrees Celsius, but normally for me, I usually use 72 degrees Celsius. Okay? And so that is optimal for TAC polymerase. All right? Ayan. So how does the PCR occur? No? Ayan. So basically, no, you have here the template strand. Ayan, you have the DNA template, no? Containing the template strands. No? This time, tatawagin ko munang template strands yung parehong strand. Okay? Ayan. Now, let's suppose meron akong gustong gene na i-amplify. No? Ayan, kunwari, no? I find this flower really beautiful, no? Kasi yung color niya ay napakaganda, no? Kunwari, it has an iridescent color. And then I was able to identify the gene responsible. Ayan. Then I was able to identify the gene that was responsible for that iridescent color ng flower, no? So pag na-identify ko na yon, what I will do is I will now I will design primers specific to the gene. So kunwari, ito yung DNA ng flower na yon. Okay? Ito yung gene that I want to amplify. Okay? So ito siya. What I want to do is I will determine a forward primer and a backward or a reverse primer. Okay? So magsisynthesize ako ng primer. Okay? Now, yung primer na ito, these primers are the ones that I will use in my PCR. Okay? So every time that I would like to extract that gene from that plant, okay, I will always get a high quantity of the gene. Okay? Mapaparami ko yung gene. For what purpose? Kunwari, gusto ko ilipat yung gene from one organism to another. Okay? Or I just want to study the structure of the gene. Okay? So, or I want to sequence the gene. All right? So those are the usual purposes. Okay? So now I have my DNA primers. No? Take note, ang difference niya sa natural replication is that sa natural replication, the primers are RNA molecules. Sa PCR, DNA molecules. All right? Yeah, so the DNA primer will be mixed with the DNA template. Okay? The first step is denaturation. Ito yung first step. Okay? At 95 degrees Celsius. And then the second step is annealing, where the primers will anneal. Okay, so you have the forward primer and the reverse primer. So this is my forward primer. Ayan, kasi 5' to 3'. Okay, and then this is my reverse primer. Ayan, reverse primer. Ayan, pag ganon, tapos pag ganon. Alright, ayan. So, the annealing temperature depends on the length of the oligonucleotide primer and also on the GC content. Okay, of the oligonucleotide primer. Okay. Next is number 3. The third step is elongation. Okay, the DNA polymerase will then add nucleic acids, on the oligonucleotide. Okay? So magdadagdag siya until humaba siya ng humaba. Okay? Now, hahaba ito ng haba until ma-reach niya yung pinaka-end of the genome of your plant. Okay? Now, every cycle, paiksi ng paiksi yung magiging products. Okay? Until finally, no, the product will be this particular gene. Okay? Ayan. So, you would need several cycles, okay, para mabawasan siya. Kasi remember, ang nangyari dito is that dito siya mag-start, doon siya mag-end. Sobrang haba niyan. Okay? And then sa next cycle, dito, kakabitan naman to ng isa pang primer. Pabagtad naman yung galaw niya. Okay? So nababawasan siya every time, no? Nag-uulit-ulit yung PCR natin. Okay? And then finally, you will only get this gene, no, from your PCR. Okay? So again, usually 25 to 30 cycles would be required to properly amplify the gene. Okay? Ayan. So basically, no, yan lang yun naman yung PCR. Okay? Ayan. And, sino sasabihin ako? Nakalimutan ko lang. Tinatry kong ilalahanin. Okay, so, okay. Now, PCR, what is next? Okay? Ayan. So after PCR, what is next? Okay? So normally, what we do is we want to determine if we're able to amplify the gene. Okay? So suppose alam ko yung size ng aking gene. Okay? So suppose the gene is 23,000, sige, wag masyado malaki yun. 250 na lang. 250 base pairs. No? So for example, if the gene is 250 base pairs, okay, I would want to know if I was able to amplify the gene. Okay? Now, how do I detect or how do I determine, no, the product of a PCR, no? Or the product of the PCR or polymerase chain reaction. Okay? So ang gagawin ko is I will perform agarose gel electrophoresis. Mas pangit yung sulat ko ngayon because yan, dalawang computer na naka-separate na. Hindi siya naka-replicate, no? Ang dami kong devices na gumagalaw. Okay, so agarose gel electrophoresis or AGE, no? Yeah, so agarose gel electrophoresis is a way to separate DNA molecules or oligonucleotides, okay, based on their size. Okay? Ayan. So the higher, no, the higher the size or the greater the length of the nucleic acid, okay, the lower or the slower will be its migration. Okay? Mas mabagal gumalaw yung mas malaking nucleic acid or oligonucleotide. Okay? The smaller oligonucleotide will move faster or will move at a longer or at a longer distance, okay, from the gel. Okay, so normally in an agarose gel electrophoresis, no, you have here, ayan, okay, so you have the wells at the top, okay. Yung DNA natin will move from one electrode to another electrode. Okay? Ayan. So, remember na, yung longer DNA will be at the top, and then the shorter DNA will be at
Pwede
the
ng towel.
shorter DNA will have longer.
Jenny, mo na yung towel magpapakanib. Wasak naman na. Ano ginahimo? Hindi mo ang towel. Sige na. Ano niya? Ano ba yun? Oh? Ano saan?
Ayan. So, teka lang, may naka-unmute, ano? Kasi mute na. Ayaw gumana ng ano. Akala ko kasi may ano, akala ko may nag-aaway. Ayan, teka lang. May ano lang dito. Ayan, okay. Thank you, thank you. Ayan. Okay. Nag-stop ha. Hindi pa na. Alright, so what I was saying is that, no, ayan. So ulitin natin. After PCR, okay, you want to determine if you were able to amplify the proper gene. Okay? Now, to be able to determine if the proper gene has been amplified, no, you will perform agarose gel electrophoresis. No? So in agarose gel electrophoresis, your DNA, okay, will move, no, from the topmost portion to the bottom portion. Okay. So the longer DNA molecules or the longer oligonucleotides will be retarded, no? Or they will travel shorter distances compared to shorter oligonucleotides, no? Shorter oligonucleotides will travel longer distances. Okay? Kasi mas maliit sila. They are able to penetrate the matrix of the gel. Okay? Now, normally, no, we add staining dye, no, to this agarose gel electrophoresis, and that is your ethidium bromide. Yeah, so it's ethidium bromide. Okay? Yeah, so ethidium bromide will intercalate between the bases of the DNA and will allow it to fluoresce under UV light. Okay, so again, the ethidium bromide will intercalate between bases of the DNA and it will allow the DNA to fluoresce under UV light. All right? Ayan. Now, the PCR, the manual, no, the conventional PCR is quite long, no, and tedious process. Imaginein mo yon, you run 25 to 30 cycles, okay? Maiksi lang naman pala yung mga oras dito, no? So, usually 30 seconds to 1 minute lang per step, okay? But multiply that by the number of cycles. Mahaba yon, no? So normally minimum 1 hour, no, yung isang PCR run, okay? It can get as long as 3 hours, okay? Yeah. So in my experience, dahil I use a different type of PCR, it's very long, mga 3 hours. Tipong when I run PCR, lalabas muna ako, no? Kaya maglalakad-lakad, bibili ako ng kwek-kwek, no? Ayan, ginugutom ko lang kayo kasi breakfast. Ayan, so lalabas ako, bibili ako ng kwek-kwek, kakain ako ng kwek-kwek, tapos may nakita kong pansit canton. Bibili rin ako ng pansit canton, no? Tapos kakain ako. And then maglalakad ako ulit, no? Magmumuni-muni, no? Sa garden. Tapos babalik ako sa lab, hindi pa siya tapos. Okay? Ayan, so ganun yung nangyari, no? Sa PCR run. Okay? Yeah, so it's really, really long. Okay? Then after that, no? You have to prepare the gel for agarose gel electrophoresis. Okay? Kaya matagal din yon. Mga 30 minutes to 1 hour, no? The entire process, no? So sobrang tagal. So there are newer methods to PCR, okay? And that is what we call your real-time, dito tayo magsulat. That is what we call your real-time PCR. Real-time PCR is also your RT-PCR. Ayan. So ano yung pinagkaiba ng conventional PCR sa real-time PCR, no? In conventional PCR, there is no detector for the genes that are being produced every cycle, okay? In real-time PCR, no, you add a dye, no? You add a dye, usually it's cyber green. Nasulat, cyber green. So usually it's cyber green. And this dye, no, can be detected by the UV spectrometer in the instrument. Doon sa mismong thermocycler, meron siyang detector. Okay? Ayan. So i-detect niya ngayon yung presence ng cyber green. Okay? So the more cyber green absorbance, no, or actually its fluorescence that you get, okay, mas maraming genes na yung produce. Okay? Ayan. So yun lang yung ano dito, no, yung nangyayari. Alright? Yun lang pinagkaiba. Okay? So it means na it's faster. No? Kasi you don't have to perform agarose gel electrophoresis anymore to identify the band of your gene. Okay? Hindi mo na kailangan gawin yon. Okay? Kailangan mo lang tingnan kung magkakaroon ka na absorbance, no, at the cyber green wavelength. Alright? Now, bukod sa real-time PCR, probably you have heard of another type of PCR, okay, which is the RT-PCR. Ayan. So ano tong RT-PCR? This is your reverse transcriptase real-time PCR. Okay? Ayan. So Narinig na yung reverse transcriptase before, di ba? Ayan. At saka syempre, nung nag-aaral pa kayo. Reverse transcriptase is also your RNA-dependent DNA polymerase. Okay? So it is your RNA-dependent DNA polymerase. Okay? Ayan. So that is your reverse transcriptase. Okay? Now, remember that the reverse transcriptase no, um, is the one that converts your RNA to DNA. Okay, so yung RNA natin, yung RNA mako-convert into what you call the cDNA. Ayan. So ano yung letter C? C stands for complementary, no, DNA. No? Ayan. So it's the complementary DNA to your RNA. Alright? Ayan. And normally, saan natin ginagamit itong reverse transcriptase? PCR. We use this in, yan, narinig na natin to during the pandemic, okay? Which we are still now in, okay? So, di ba yung reverse transcriptase PCR ay ginagamit in the detection of your COVID-19 sa yung RT-PCR na tinatawag, di ba? Ayan, ayan, na-connect na, ayan. Okay, so this one is actually used in the detection of viral organisms. Okay, viral organisms, viral antigens, no, not organisms, and viral antigens like your COVID-19. Okay, kasi yung COVID-19 is an RNA virus. Okay, tapos to be able to identify the presence of the virus in the in your tissue. Okay, so kailangan mo lang no i-convert yung RNA to DNA and then i-determine mo kung siya nga ba ay galing sa COVID-19 virus. Okay? Sa SARS-CoV-2. Alright? Ayan. So, that is the importance of your reverse transcriptase real-time PCR. Okay? So, ang pinagkaiba lang is the DNA polymerase, no? Bali, before you perform the PCR, no? The real-time PCR, before you do that, okay, you have to convert first your RNA to DNA using a reverse transcriptase. Okay? After nun, pag meron na akong cDNA, kaya ko nang paulit-ulitin yung cDNA ko using the conventional or the RT-PCR. Okay? Ayan. So yung reverse transcriptase real-time PCR, ganito lang yung nangyayari doon. First, you need to convert your RNA to cDNA using a reverse transcriptase. Okay? And then, yung complementary DNA, i-replicate natin, paparamihin lang natin to determine no, the presence of the gene. Okay? Ayan. Of course, ang ginagamit na sa most laboratories ngayon ay real-time PCR. Kaya nga sinasabi nila, di ba, within 30 minutes to 1 hour, no, pwede ka nang mayroong result. Okay? Yeah, so pwede rin siyang gamitin for the detection of retroviruses. Okay? Kasi retroviruses ay RNA viruses din. Okay? So like yung HIV. Okay? Ayan. So yan yung tungkol sa PCR. Now let us proceed to the next one. Ayan. Another method of increasing the quantities of the gene, no, is molecular cloning. Although this one is much older, okay? Kaya mas luma ito, okay? So kung yung PCR ay in vitro amplification of gene sequences, your molecular cloning is in vivo, no? Ayan, pinapasok natin, no? We insert the gene in a plasmid and then we replicate the plasmid by growing your bacteria or your fungi. Okay? Yan. So that is your molecular cloning. Okay? So para lang mas maintindihan natin maigi, no? We know that bacteria, no, ay mabilis mag-grow. Okay? Syempre compared to human cells, fungal cells are also fast to grow, no? For example, yeast cells, no, mabilis din sila mag-grow. Okay? Yan. So you can use those cells, no, the bacterial cells or yeast, no, to amplify the gene. Okay? So how do we then amplify the gene? Ito yung tinatawag natin na cut and paste method. Okay? Yan, cut and paste naman. Ayan. So pag nakita niyo yung term na cut and paste, ito na yun. So for example, we have a gene. Ganun ulit, no? Mag-drawing ako. May kinatutuan akong gene. May kinatutuan akong gene. Kunwari yung gene nga dun sa flower ng ganda ng kulay, iridescent.
Sir,
Okay?
excuse me
Ito
po.
yun. Okay.
Sir.
Yan. So what I can do is I
Sir?
can cut the gene. No? Ayan,
Sir,
ika-cut ko
excuse
siya.
me po.
I cut ko yung gene over here and over here. Okay? So what do I do to cut the gene? So normally, no, I would look for sequences that are amenable to cutting. Yung pwede kong
Sir?
gupitin. Okay? And usually
Sir?
what I will use ay yung tinatawag nating restriction
Sir?
endonucleases. Yes? May
Hindi po
nagre-
nakikita yung share screen niyo po, sir. Nawala po ata.
Ay, hindi nakikita yung share screen. I'm so sorry. Akala ko kasi may nag-aaway ulit doon sa online world. Okay, yeah. So ito lang naman, no? Yan pa lang naman tayo. Okay. So, another technique is molecular cloning. This is your cut and paste method, no? So I have a gene, you look for regions in the gene that you can cut, okay? You cut it using restriction endonucleases. Okay? Ayan. Now, restriction endonucleases are enzymes that search for restriction sites and recognition sites. Okay? There are some restriction endonucleases na same lang yung restriction site and yung recognition site. Okay? But there are also endonucleases na magkaiba yung restriction site doon sa recognition site. Okay? Respectively, they are called your type 1 and type 2 restriction endonucleases. Okay? Gets? Ayan. They are also known as your restriction enzymes. Okay? Restriction endonucleases or restriction enzymes. Okay? So, mas malinaw lang yung term na endonucleases. Okay? So, pag kinap ko na yung gene, ano yung next na gagawin ko, no? Once I cut the gene, I would transfer it to a plasmid. Okay? Since this is molecular cloning, ilalagay ko siya sa isang cloning plasmid. Now, what is a plasmid? A plasmid is a circular DNA, no, that is replicated, no, conservatively in a bacterial cell. Okay? Ayan. Okay, so for example, yung bacterial cell, meron siyang dalawang types ng may dalawa siyang types ng DNA, no? So meron siyang plasmid DNA which is circular, okay? And meron siyang chromosomal or genomic DNA na tinatawag na, ay not chromosomal, tinatawag nating genomic DNA, okay? You have the plasmid DNA, yung circular, and the genomic DNA, all right? So normally, we insert the gene in a plasmid, okay? So gugupitin ko rin yung plasmid. Now, yung plasmid na to, dapat gugupitin ko at the same site kung saan ko ginupit yung aking gene. Okay? Parang puzzle piece. Okay? Ayan. So, they should have the same ends. Okay? Ayan. And then, I will transfer the gene, ipipaste ko siya, using DNA ligase. Okay? Ayan. So, the enzyme that is used to paste the gene, no, to a plasmid that has been opened by the restriction endonuclease ay tinatawag nating DNA ligase. Alright? Ayan. So, remember din na yung pangbukas ninyo ng plasmid dapat ay same restriction endonuclease as you use doon sa gene para makuha mo siya. Okay? Yan, para compatible silang dalawa. Okay? And then, you insert the gene in your cloning vector. Okay? This is the plasmid. No? Tinatawag rin natin siyang cloning vector. Okay? Kasi tagadala siya. No? Tagadala. It's a vector, it's a vehicle. No? For the clones. Ayan. And then, next is you will insert the recombinant DNA in a cell. Usually, bacterial cell. No? Ayan. So, kunwari ito, bacterial cell, ipapasok mo siya, no, para magkaroon siya ng new gene. Okay? And then, you allow the bacteria to grow. Okay? So, dadami yung bacteria. After mag-grow ng bacteria, dumami na siya, you extract the DNA. Okay? So, extract mo yung plasmid. Ayan, kukunin mo yung plasmid. This time, it's already recombinant. Tapos, you can then cut it again. Ayan. Tapos, pwede mo na siyang i-agarose gel electrophoresis ulit. Alright? To identify the gene. Okay? Or sometimes you can immediately perform agarose gel electrophoresis on the plasmid. para malaman mo kung successful yung insertion, dapat mas mahaba yung or mas malaki yung plasmid, okay, by the size of your gene. Okay? But there are other ways, no, to determine insertion of the gene, okay? Pero masyado na siyang komplikado, no? Yan, meron kasi tayong tinatawag na LAC-Z, no? LAC-Z indicator, no? And meron din tayong tinatawag na blue and white screening, no, to determine the successful insertion of the genes. Okay? Ayan. So just take note of those terms. Now, the method of inserting the plasmid, the recombinant plasmid into the cell, no, is usually heat shock transformation. Okay? Although there are many other ways, no? This is just the more common. Okay? This is the more common method of inserting the gene into the bacterial cell. Ayan. So basically, all the reactions that I mentioned are in this slide, no? Ayan. As you can see, you have your vector, which is basically your plasmid. Hindi nga pala nakikita ang ano dito. Ayan. So ito yung plasmid, and then you cut your plasmid, you insert the DNA, okay? Then you use DNA ligase to connect or paste the DNA into the plasmid. Ayan. And then you plate, no? You allow the bacteria to grow. Okay? Yan. Normally, ang ginagamit din natin pala, no, pang-select kung na-insert ba natin siya or hindi, ay yung tinatawag natin na mga antimicrobial resistance genes. Okay? Ayan. So, pag properly inserted yung recombinant plasmid in the bacteria, dapat mag-grow siya in a plate containing the antimicrobial agent kung saan, no, susceptible yung bacteria. For example, E. coli, no? So E. coli is susceptible to ampicillin. Okay? Ayan. Ngayon, kapag successful yung insertion mo ng isang recombinant gene, okay? Usually ang ginagawa natin, yung recombinant gene, lalagyan natin siya ng antimicrobial resistance, no? Ng ampicillin resistance, for example. So pag nabuhay, no, pag nabuhay yung bacteria doon sa plate na mayroong ampicillin, ibig sabihin na insert natin yung gene. Okay? Yan. So kaya niya na ngayong mabuhay doon sa plate containing the ampicillin. Okay? So that's one way. Okay? Meron pang iba pa, no? Ayan, na pang-determine ng proper insertion of your plasmid and the gene in the bacterial cell. So shown on the right side naman, ito, ayan, these are just examples of what you call your restriction endonucleases. Okay? Ayan, so pinapakita lang where the cleavages, no, happen, okay? So kung saan nakakat yung DNA natin. Meron tayong tinatawag na blunt ends, no? Ayan. So ito yung nagfo-form ng blunt ends. Ayan. So ano ang ibig sabihin ng blunt ends? N yan, N. Blunt, hindi yan M, no? N, nanay. Ayan. So blunt ends siya kasi yung pag-cleave niya is pag ganito, straight. Okay? Blunt ends. Okay? Mahirap ikabit ito, no? Yan. So, isa sa mga mahirap ikabit ito, no? Yung blunt ends. Alright? Now, ang pinipili natin kadalasan ay ito. Yung tinatawag natin na mayroong sticky ends. So, sticky ends, ang ibig sabihin lang, no? Meron kang overhangs. Okay? May mga sobra. Okay? So, imagine niyo kapag hinati ko to, ganito siya. Yan. This is the overhang. This is also an overhang. Okay? Kaya siya sticky ends. Okay? Kaya so kumbaga pag naggaano tayo, no, nagka-carpentry, 'di ba? Naglalagay tayo kadalasan ng pagganon, no? Pagdidikitan. Okay? So yun yung overhangs. Okay? Meron tayong 5' overhang and meron tayong 3' overhang. So this one ay merong 3' overhang. This one, 3' overhang yan. And this one is a 5' overhang. Okay? Ayan. So, yung BAM-H1, 3' overhang, HIND3 is 3' overhang. Ay, 5' overhang. So, nabaliktad kaya. BAM-H1 is 3', and then HIND3 is 5' overhang. Okay? Ayan. So, yan lang naman. This one is also a 5' overhang. So, like eco R1. Masyado na specific yan, kahit na hindi na, huwag nyo na masyadong pansinin. Okay. Now, what I have just mentioned ay uh, cloning, no? Ayan. So remember, pag sinabing molecular cloning, it is just an in vivo method, no, of amplifying the gene. Okay? Ayan, in vivo method of amplifying the gene. Now, if you want to express the protein, no, kung gusto mong ma-convert yung gene into its gene product, usually a protein, okay, you need to place the gene in an expression vector. Okay? Yeah, so magkaiba yung dalawang yon, no? So the plasmid that we use for molecular cloning is called cloning vector. Okay? Tapos the plasmid that we use to express, no, the gene product is called an expression vector, no? Ayan. Pero same lang halos yung methods. Alright? Ayan. But when it comes to the expression vector, medyo namimili tayo, no, ng tamang expression system. Okay? So normally, for eukaryotic genes, we express them in fungal cells. Okay? Ayan. So for eukaryotic genes, usually we want to express them in eukaryotic cells. Okay? Not bacterial cells. Okay? Yan, kasi walang post-translational modification sa bacterial cells. Alright? Ayan. Now, meron pa ako isa pang idadagdag na wala dito, no? Another technique which is very important for analysis of DNA, no, is called southern blotting. Ayan. So, may, may ano, para akala ko may airport na dito. Ganun. Ayan, so meron din tayo, aside from agarose gel electrophoresis, no, we also have this technique, no, which is called southern blot. Okay? So sa southern blot, no, it's basically similar to agarose gel electrophoresis. But instead of using agarose gel, no, as your as the medium where the DNA will flow through, no, we use a polymeric gel. Okay? So gumagamit tayo ng polymeric gel. Okay? Ganun pa rin naman. The larger DNA will not move too far. The shorter DNA will move farther. Okay? Yan. So pareho lang ng agarose gel electrophoresis. However, meron pa tayong iba pang ginagawa. In southern blot, no, what we do is we normally identify the DNA using either an antibody, no, and specific to that gene, okay, or we transfer the DNA onto another gel, okay, kung saan we stain the DNA with a dye. Okay? Kaya siya tinatawag na southern blot. Okay? So binablot natin, di ba? Pag sinabing blot, tinatransfer, dinidikit, di ba? Ayan. So southern blot. So remember that southern blot is used for the analysis of DNA. Okay? And then meron pang isa, northern blot. Ayan. So northern blot naman is used for RNA. Okay? Ayan. So remember these two terms. Actually, may pangatlo pa. Okay? Pero wala pa kasi tayo doon. Pero sige, sabihin ko na lang din. No? Baka makalimutan ko pa mamaya. The third one is western blot. Ayan. Ayan. So western blot is for proteins. Okay, yan. So yan yung tandaan nyo, no? Yung table na to. Okay? Ayan, no. Actually, may kwento to na nakakatawa. Ewan ko kung matatawa kayo. Kasi, the reason why it's called southern blot, kasi yung naka-discover or naka-invento sa kanya ay may surname na southern. Okay? Ayan. Tapos, ngayon, dahil nga naka-invento sila ng iba pang methods, okay, related to southern blot, no? Ayan, pero ibang biomolecule lang yung dinedetect, okay? Sinamahan na lang nila ng directional na words, no? So, 'di ba, may southern na based on a surname, tapos nilagyan nila ng northern, okay? Tapos may western, okay? Ayan. For fun lang. Ganun yung mga molecular biologists, no? Ayan, minsan nagtitrip sila sa mga pangalan, no? ng mga bagay-bagay. For example, yung CRISPR-Cas9, no? Familiar with CRISPR-Cas9, di ba? Parang ang daling tandaan kasi, no? CRISPR, no? Ayan. So it's a method of gene editing, no? Ayan. So CRISPR-Cas9. Okay? Bago yun ngayon. Ayan. Okay, so lilipat na ako ulit. Ayan, so bago ko makalimutan, ayan, mag-share screen po ako. Ayan, okay. Okay, so the next one is not hindi siya gumagal. Ayan. Now let's move on to the other nucleic acids, no? So this time, let's talk about your RNA. Medyo maiksi na lang yung part na ito, no? So na-mention na natin to before, no? You have the rRNA, the mRNA, and the tRNA, okay? So rRNA is your ribosomal RNA, tapos your tRNA is your transfer RNA, and then your mRNA is your messenger RNA, okay? So it is important for you to remember what they are used for. Okay? So yung rRNA, the ribosomal RNA, no, is the RNA that is associated with the ribosome, which is the macromolecular complex containing proteins and the RNA. Okay? So yun yung ribosomal RNA. Okay? Now remember then, no, yung pinag-usapan natin before, that prokaryotes and eukaryotes have different sizes of your ribosomal RNAs, di ba? Yung prokaryotic RNA ay 70S, no? And then yung eukaryotic naman na rRNA ay 80S. Okay? Now there are two subunits, no, in the ribosomal RNA, the smaller subunit and the larger subunit. For the prokaryotic RNA, no, the smaller subunit is 30S and the larger subunit is 50S. Okay? For prokaryotics. For prokaryotes. Now for eukaryotes, no, for eukaryotes, the smaller subunit is 40S and then the larger subunit is 60S. Okay? Now, in fact, no, yung mga ribosomal subunits na iyon ay ito. No? Your 30S ribosomal subunit and your 50S ribosomal subunit would comprise these, and your 40S and 60S ribosomal subunit will comprise these ribosomes, ribosomal RNAs. Okay. Now, S stands for Svesberg units. Yeah, so take note of that term. No, Svedberg. Yeah, units. Yeah, Svedberg units, no? Svedberg units refer to the sedimentation rate, no, of the molecular complex. Okay? So the larger the number of the Svedberg unit, the faster is its sedimentation. Okay? Ayan. Kasi nga bumabagsak siya. Okay? Nang mas mabilis kaysa dun sa mga smaller na subunits. Okay? So related yang Svedberg unit sa sedimentation rate of the macromolecular complex. Okay? Now, when you combine the larger and the smaller ribosomal subunits, nagbabago yung total, okay, ng Svedberg unit, di ba? Yun yung sinabi ko kahapon, no? Yung 30S plus 50S ay equal to 70S, no? Yan. Di ba, ang labo. Tapos yung 40S plus 60S ay equal to 80S, no? Ayan. So bakit nagbabago yung total? Kasi when you have a macromolecular complex, no, there are areas, no, yung areas na concealed, no? Ayan. So 'di ba pag nagdikit na sila, yung total surface area, no, yung sum of the surface area of the two ribosomal subunits, okay, will not be equal to the total surface area of the whole unit, 'di ba? Ayan, parang ganito yung sinasabi ko. You have the smaller subunit and you have the larger subunit. Pag kinuha mo yung surface area nito plus surface area na ito, no, that's actually definitely larger compared sa kapag pinagsama ko na yung dalawa. Okay, ayan. Yung surface area na ito. Okay? So, mas maliit yung surface area pag combined na sila. Okay? Kaya mas maliit talaga yung Svedberg unit of the macromolecular complex combining the two. Okay? Ayan. So, yan lang naman yun. Baka lang na-curious kayo. Next is the tRNA. No? So the tRNA is the transfer RNA. No? Its use is as an adapter molecule. No? It carries with it the anticodons to the codons present in the mRNA. No? And the tRNA also carries with it the amino acids that will be added in the peptide chain. Okay? Ayan. So, yan yung purpose ng ating transfer RNA. Okay, so next, we have the messenger RNA. So the messenger RNA is your transcript, no? Ayan. So it is the encoded transcript from the DNA, no? And it usually contains, no, the sequence of nucleic acids that will be used, no, that will be used as a code, no, for certain amino acids, no, for the synthesis of proteins. Okay? Ayan. So in the mRNA, okay, take note na the mRNA, no, needs to be stable. Okay, it needs to be stable. Kaya you have certain modifications like the guanosine cap, no, the guanosine cap, and ito yon. And then you have here the polyadenylate tail. Okay, ayan. So these modifications actually enhance, no, the stability of the mRNA. No? Kasi remember that RNAs are less stable compared to DNA. Okay, yeah. So lagi yun yung common, no? Na characteristic of RNAs, no? They are less stable compared to your DNA. Na madali silang ma-degrade. Okay? Actually, ang joke namin sa lab, no? Yung RNA, maalog mo lang yung sample mo, no? Sira na yung RNA. Okay? Kaya nga pag nagte-testing, no, ng mga COVID, yung sa COVID-19, no, pag sinabing shake by turning your tube upside up and down for 3 times lang, sundin mo yun, no? Yan. Kasi pag ang ginawa mo, inalug-alug mo siyang ganyan, no? Yung RNA na laman, no, ng inyong sample, masisira. Okay? Yeah, so akala mo wala kang COVID-19 pero meron pala, no? Kasi nasira lang pala yung RNA. Okay? Yeah. So, yun, very unstable yung RNA. So, to stabilize, no, in the body, the RNA, there must be the methylguanosine cap and the polyadenylate tail. Now, remember that not all areas or regions in the mRNA ay coding regions. No? There are also yung tinatawag natin na 5' and 3' untranslated region. Okay, so ito yung tinatawag natin UTR. And UTR or untranslated region. Yan, pwede siyang 3' or 5'. Ayan. Now, let's talk about the initiation. I will not dive very deep in transcription kasi medyo complicated siya. Ito lang yung gusto kong i-highlight. No? Always remember that the initiator sequence, no, in prokaryotes and eukaryotes are different. Okay? For the prokaryotes, it is the -35 sequence and the Pribno box. Okay? Yan. So yan yung tinatawag natin na promoter region. Okay? Anong ibig sabihin itong promoter region? That is where your transcription factor will bind to. Okay? Yan, doon siya didikit. Okay? Para ma-initiate yung transcription. Okay? Yan yung sa prokaryotes. -35 sequence and the Pribno box. Now remember that this sequence ay consensus sequence, no? Yan, or it's a conserved sequence, no? Ang ibig sabihin ng conserved sequence, all bacteria will have this sequence, no? Kasi this determines where to start the transcription. For eukaryotes, no, it's the tata box and the kaat box. No? Ayan. So para madaling tandaan, pag tunog pangalan, okay, si kat at si tata, no? Ayan, yun yung sa eukaryotes, no? Kaat box and tata box, no? So tata is also the Hogness box. All right? Ayan. Now, after the initiation, of course, there will be the elongation, no? Kung saan napoproduce yung ating mRNA transcript. Now, remember, in producing the mRNA, no, napapalitan yung kapartner ng inyong adenine. Okay? So remember that there is no thymine in the RNA. No? There is no thymine in the RNA. So sino yung pumapalit sa kanya? Your uracil. Okay? Ayan. So for example, I have here a sequence of DNA. So kunwari may sequence ako ng DNA na 3' to 5'. So ganito yung sequence niya. CAT GAC. Okay? Ayan. So for example, I have this DNA, okay? When it is converted into an mRNA transcript, no? Ayan. Take note that the mRNA Take note that the mRNA is elongated also from 5' to 3'. No? Ayan. Para madaling tandaan, lahat 5' to 3'. Okay? Ayan. So, yung mRNA mo ay maggagalaw from 5' to 3'. Yung kulay red, that's your mRNA. Okay? So, what will be the complementary strand, no, of this DNA? Okay? Ayan. So, ang partner ng C ay G. Okay? Partner ng A ay U, no? Kasi walang T ang RNA, no? So this will be U. Tapos T, sino partner ng T? This is A, adenine. Okay? And then sino yung partner ng G? C. Sino yung partner ng A? U ulit. No? Ayan. So kasi nga walang thymine, no? Ayan. So laging ATGC, no? Except in the RNA where you have AU. Okay? And then finally C, ang partner niya ay G. Okay? So if your DNA, lagyan natin ng marker. If your DNA is CATGAC or in the proper sequence, no, ang basa natin talaga sa DNA na to ay CAGTAC. Okay? Yan. Gets? Yan. So ang mRNA na mapoproduce niya is GUACUG. Alright? Ayan. Kasi 5' to 3'. Alright? Ayan. So if I give you the entire, so if I give you the entire DNA duplex, no? Kunwari naglagay ako ng isa pang DNA duplex, no? Ayan. So lagyan ko ng G, T, A, C, T, G. Okay? This one is your 5' to 3'. Okay? And I have to mention which one is the template DNA. Okay? Ayan. So yung template, kailangan ko sabihin na ito yung template DNA and the other one is the non-template DNA. Okay? Ayan. So what you will transcribe is the template DNA and not the non-template DNA. Alright? Ayan. Saan tandaan nyo lang, 5' to 3' palagi. Ayan. Okay? Clear tayo doon? Okay? So, tsaka walang T sa RNA. No? Only U. Hey, may isa pa pala. Now, how do we end the transcription? Meron tayong tinatawag na termination. Okay? Now, there are two types of termination. Meron tayong tinatawag na raw dependent, no? And the raw independent. No? Raw dependent and raw independent termination. Okay? So, yung raw, it's a protein. Okay? So, it's a protein that signals, hoy, stop na. Okay, yan. Stop ng pagkopya, no? Ayan. So, parang pag nag-ano, no? Pag nag-exam, no, tapos yung teacher mo naglilibot, tapos nakita mo, may nangongopya. Hoy, stop na yung pagkakopya, no? Ayan. So yan yung raw protein, okay? Now, if there is no raw protein, no, what happens is that your RNA becomes a hairpin. Okay? Nagkakaroon siya ng hairpin sa dulo, and then that will signal your transcription factor to stop. Okay? So yun yung tinatawag natin na raw independent termination. Okay? Ayan. So ito yung raw independent, no? May hairpin na napoproduce. Okay? Ayan. That will signal the stop of your transcription. Alright? Raw. Ayan. RHO. Ayan. Para iba yata yung spelling. Yeah. So, tamang hindi po nakapansin, nandito yung raw. So, okay. Buti, dapat sinasabi nyo kasi sa akin, no? Hindi yung ano, nagtatahimik lang kayo dyan. Okay. Ayan. Now, remember, no, na the eukaryotic, no, mRNA requires processing. Diba? In-explain ko to before. Meron tayong tinatawag na splicing, no? And the splicing happens in the presence of your spliceosome. Okay? And the splicing basically removes the intervening sequences or the introns. No? Ayan. So yung introns, ayan, usually appear interspersed or in between of your exons. No? Ayan. Pag sinabing exons, ito yung naiiwan after splicing. Okay? So, drawing tayo kunwari. Yan. So, let's suppose this is an exon. Yan. So, bawat box ay exon. Yan. Binaliktad ko yung drawing ko kasi yung sana una kahapon, yung nakabox yung intron, no? This time ang nakabox yung exon. Yan. So, during splicing, ang nangyayari is natatanggal itong mga introns. Matatanggal ito, matatanggal ito, matatanggal ito. Okay? And then the exons will be combined. Okay? Yan. So, magdidikit ngayon yung inyong exons. Yan. So, you have now a mature mRNA. Okay, so this is your pre-mRNA, and then this is your mature RNA. Mature mRNA. Okay? And so remember that kadalasan it uses spliceosome, and the process is called splicing. Okay? This only happens in eukaryotic mRNA. Okay? Yan. Sa prokaryotic mRNA, remember, walang processing kasi sabay-sabay nangyayari or pwedeng nangyayari ang replication, transcription, and even translation. Okay? Yan. This one is fun. Okay? So, codons. No? So, we are now in translation. No? Nandun na tayo sa last step in the central dogma. Okay? Huwag niyong i-memorize to. So, hindi ko rin memorize yan. No? So you have here the table of codons. Okay. Now, what are codons? Okay. Codons are the three letter or the three base patterns, no, that are identified by the tRNA, no, carrying a particular amino acid. Okay. To incorporate in the growing peptide chain. Okay. So basically, yung codons, no, ito yung code, no, it is the code for an amino acid. Okay? Ayan. So, yun lang naman, no? It's a three-letter code, no? For an amino acid. Okay? Now, remember that codons, no, have the following characteristics, no? Explain ko silang lahat. One is universal. So, pag sinabing universal, it means it occurs, no, in all organisms, no? From prokaryotes to eukaryotes. From bacteria to protists, to plants, to animals, no, to fungi, to all organisms. Okay? Universal siya. Okay? Even though it is universal, meron tayong tinatawag na codon usage, no? Ayan. So ibig sabihin, certain organisms would prefer, no, one amino acid over the other. Okay? But still the code will be the same, no? Pare-pareho sila ng template, no? Kahit na anong organism pa yan, no? It's universal. Number two, it's specific or unambiguous, no? So ano yung ibig sabihin natin ng specific or unambiguous? It means na one codon is to one amino acid and not more. Okay? So one codon, okay? One codon means one amino acid or is related to one amino acid only. Okay? Ayan. Anong ibig sabihin natin nun? To read the codon, okay? So kailangan nating gamitin itong table na ito. Remember that we read, no? We translate also from 5' to 3'. Okay? Ayan. So 5', 3'. Okay? So we have the first, we have here the first base, tapos the second base and the third base. Okay? So for example, pili ako, no? So kunwari gawa tayo ng ng code. CUA, no? Ayan. So CUA. Ayan. So hanapin natin si CUA, no? Ayan. So ito yung C, okay? Andito yung U, okay? Ayan, nakikita, no? So CU, tapos yung A, ito, that's leucine. So CUA is only leucine, nothing more. Okay? Okay? Ayan. Pili ako, isip tayo ng iba pa. Kunwari, GAA na lang. No? GAA. Ayan. So GAA. Ayan. So hanapin natin si GAA. So this is G, this is A. Okay? And then, A ulit. So, that is your glutamate. May GLU, glutamate. Okay? Ayan. So, isa lang, no? One codon refers to or is encoded into one amino acid only. That is what you mean by specific or unambiguous. Ibig sabihin, yung isang codon, hindi siya more than one amino acid. Okay? Ayan. So, 1:1. One codon is one amino acid. Alright? Ito yung pang-confuse. Number 3, degenerate. Okay? So, the codon is degenerate. No? So, anong ibig sabihin nito? Ayan. Ang ibig sabihin naman ng degeneracy ay ito. One amino acid, 1AA, is encoded by possibly multiple codons. Ang gulo, no? Ayan. Sabi natin unambiguous, no? One codon is to one amino acid. Pero degenerate yung ating codon. One amino acid can be encoded by multiple codons. Okay? Anong ibig sabihin natin doon? Tingnan natin yung leucine since na-identify na natin siya kanina. For leucine, ayan. For leucine, you will see na ang dami nun. Ito yung lahat. Oh. Ayan, leucine yan lahat. Okay? So, gaano karaming codons yung pwede nating isulat, no, that will produce leucine? Marami, no? Yeah, so, pwedeng CUA, mali pala, ito pala yung CUA kanina, no? Sorry, namali ako doon. Ayan, burahin natin ito. Ayan, CCA yung kanina. So, pwedeng CUA, ayan, so CUA, okay? Pwedeng CUG, CUG, pwedeng CUU, tapos pwedeng CUC, ayan, pwedeng CUA, ano pa ba? CUA, yeah, yun na nga yun. Tapos CUG, yun na yun, okay? Yeah, so there are several codons, no, that will encode the same amino acid. And it is said to be degenerate. Why is that so? No? Bakit kailangan siya maging degenerate? No? Ganito yung purpose niya. No? Codons are degenerate because we want to prevent mutations. Okay? So for example, if there would be a mutation in one nucleotide, okay, tapos mapapalitan agad yung amino acid, edi masisira na agad yung protein. Then the function may be altered already. Okay? Pero kapag multiple codons could encode the same amino acid, even if there will be mutation, okay, even if there will be mutation in the, even though there will be mutation in the, even though there will be mutation in the sequence, okay, it could still produce the same amino acid. And that is what you call silent mutation. Yeah. Actually, no, bigla ko na-realize na iba tinitingnan kong letter on the side. So, ulitin natin yung kanina na. No? So, leucine is UUA, UUG, ayan, UUA, UUG, ayan, so UUA, UUG, CUU, CUC, CUA, CUG. Alright? Ayan. Okay? Ayan. So, ganun yung pagbasa. Na-realize ko lang, sabi ko, may yung nga pala dun sa taas. Okay? Ayan. So, mahirap nang malabo yung mata. Okay. So, ang tawag natin, no, sa mutation that does not alter the amino acid sequence is silent mutation. Okay? Ayan. So, yan yung kagandahan ng degeneracy of the codons. Gets? Ayan. So, aside from leucine, of course, we have others, no? You like, for example, si proline, si serine, no? They are encoded by different codons. Okay? Gets? Ayan. Okay na? Ayan. Sorry dun sa confusion kanina. Okay? Ayan. And then lastly, we have non-overlapping and cameless. No? So, anong ibig sabihin nitong non-overlapping and cameless? So, for example, I have a sequence na CUG, UA, C, ayan, tapos AAC. Okay? So it is non-overlapping and it is commaless. So ibig sabihin, pag binasa siya, pag ganon, and pag ganon. Okay? Hindi siya tumatalon, hindi rin siya nag-overlap, no? So, it means na hindi siya ganito, no? 1, tapos biglang umatras, no? Ganyan. 2, tapos umatras ulit, no? 3, okay? Hindi ganyan, okay? It is non-overlapping and it is comma-less. Alright? Yan. However, ang tawag natin doon sa area kung na na-encode ay ang tawag natin sa area na i-encode in your mRNA is the reading frame. So this is what you call the reading frame. Ayan. Okay? Yeah, now, how does protein synthesis occurs? Ayan. How does protein synthesis occur? Ayan. So basically, no, summarize natin. Yung tRNA, may bit-bit siya. Okay? So yung tRNA, may bit-bit siya na amino acid. Okay? And then, papasok siya sa inyong ribosome. But before I forget, may nakalimutan akong banggitin pala dito sa kabila. Okay? Remember these 4 codons. Okay? Ayan. So si AUG, okay? So hanapin natin si AUG. AUG. Okay? This is methionine. Okay? AUG is your start codon. Okay? So AUG is the start codon. All right? Next. Yung tatlong dapat yung i-memorize ay si UAA, UAG, and UGA. Ayan. Ayan. So, isipin nyo lang, no, na para kayong unggoy, no, na nagsasalita, no? UAA, UAG, UGA, no? Ayan. So, so yung tatlong yan, no, UAA, UAG, UGA, no? Ayan. So, pwede mo rin balibalik na rin. UGA, UAA, no? Ayan. So, those 3 codons are your stop codons. Okay? Ayan. So, stop. So, ibig sabihin, pag nakita yan ng inyong tRNA, no? Stop, no? Hindi na magko-continue yung translation. Alright? Sabi nga nila, no, just in case makalimutan niyo yung start codon, no, since yung August daw yung start, no, nakita ko lang yung meme sa Facebook recently, kaya daw AUG ang August kasi it's the start of classes, no, ayan. Diba? Ayan, so, ayan. So, although, pag nakausap mo yung mga tao from the 90s, no, mapapaisip sila kasi yung mga tao ng 90s, ang start ng classes June, no. Ayan. Pero sa mga kabataan ngayon, ang alam nilang start of classes August, no, AUG, start codon. Di ba? Nakita ko lang yan sa isang meme lately. Ayan. All right. Now, what happens during translation? So remember, your start codon is your AUG. Okay? So the anticodon, no, the anticodon of the start codon, AUG, no, can be found, no, in your initiator tRNA, no, which actually carries with it, no, the methionine amino acid. Okay? Now, the methionine amino acid is first activated to form N-formyl methionine, okay, through a transformylase reaction. Now, this tRNA can then enter the A site, no, the A site of your ribosome, okay, doon siya papasok, okay, and then mayroon ng susunod sa kanya na isa pang tRNA, no? So, let's say yung another tRNA na yun has this anticodon, ACG, which carries with it the cysteine, no? The cysteine amino acid. This one will then enter the A site also. Pag pumasok sa A site, yung pangalawang tRNA carrying an amino acid, the first amino acid na nasa A site ay magta-transfer sa P site, no? A stands for amino acyl site. Okay? Tapos P is the peptidyl site. No? Ayan, peptidyl, P, no? Bakit tinatawag na peptidyl site yung P site, no? Kasi imagine niyo to, you have, let's say here, lysine and valine. Okay? So yung bagong pasok na tRNA carries lysine. Okay? And then yung nauna sa kanya is valine. Okay? So the peptide bond is formed in your peptidyl site. Okay? Yan. So dito siya na-form. Okay? Kaya siya tinatawag na P site. Okay? And then magmo-move yung valine papunta sa E site. Okay? That's your exit site. So lalabas na siya. Alright? So basically, ganyan yung translation. No? So again, simulan natin sa simula. You have the start codon AUG, no? The tRNA carrying methionine will proceed to that region where you have the start codon AUG. Okay? However, before that happens, your methionine must be activated to form formyl methionine. Okay? Tapos, darating yung isa pang tRNA, okay, carrying with it an amino acid, papasok sa A site, your methionine will move to the P site. The peptide bond between methionine and the next amino acid will be formed. Okay? Darating yung susunod, yung pangatlong amino acid, papasok sa A site, yung nasa A site, lilipat sa P site, yung nasa P site, lilipat sa E site, exit. Okay? And then, the process will continue. Okay? That's basically your translation. Okay? Just remember, APE, amino acyl site, peptidyl site, and exit site. Okay? Ayan. So, yan yung translation. Ayan. Huwag na yung part na yan. Okay, so this is another diagram no, showing to you how the peptide bonds form between the two amino acids. Ayan. Hindi naman masyadong important ito. Okay, now let's talk about mutation. Ayan. So, mutation is an inheritable change or variation in the gene. No? Ayan. When I was younger, okay, nung high school kami, my teacher asked us, no, sabi niya, biology teacher, first year high school to. Sabi ng teacher namin, wala kaming grade 7 noon. So, yung first year high school ko ay after grade 6. Ayan. So, nagtanong yung teacher ko, who here wants to be mutants? Kasi ang pinag-uusapan namin doon, yung Chernobyl accident. No? Ayan, familiar with the Chernobyl accident? Yung sa Russia, may sumabog na nuclear, no? Power plant, okay? Tapos nag-shower, no, ng radioactive materials sa paligid ng Chernobyl, no? So maraming tao na naapektuhan, no? They actually deserted the area, no? Hanggang ngayon, there are still radioactive isotopes in that area, no? Hindi pa rin siya pinupuntahan. Kung nanonood din kayo ng Marvel, no? Ayan. So, yung mga scrolls, okay? Yung mga scrolls sa Secret Invasion, okay? They are able to withstand daw high levels of radiation. Kaya sila tumira sa isang abandoned power plant, no? Nuclear power plant. Ayun. So, let's go back. So, ayun yung pinag-uusapan namin, no? Yung Chernobyl accident. So, my teacher asked, sabi niya, who here wants to be mutant? No? Ayan. Kasi nagtaas ako ng kamay. No? Ayan. Tapos, nung time na yon, no? Nagtataka ako, ba't walang ibang nagtataas ng kamay? Ba't ako lang? Tapos ang saya-saya ko pa, no? Doon sa sagot ko na I want to be a mutant, no? Kasi I was watching X-Men, no? Ayan. kasi yung mga X-Men kasi, they are mutants, no? And gusto ko magkaroon ng superpowers, no? Kaya nag-raise ako ng hand, no? Tapos parang tinawanan ako ng buong klase, no? Kasi ako lang pala hindi nakaintindi, no? Ayan. Then that's now a learning moment for me, no? It's not good to be mutant, no? Unless you are in the comics, okay? Ayan. So now mutants, no? Ayan, basically have this gene that has been altered already, and it can be transferred from one generation to the next, no? So that is what you call mutation. Now, those with the normal gene ay tinatawag natin na wild type. Okay? Ayan. So kung meron tayong wild type, ito yung normal, okay? E meron tayong mutant type, no? Ito naman yung mayroong mutant na gene. Alright? Ayan. And it's not always good to be mutant, no? Ayan. So, parang very rare, no, na yung mutation ay maganda. Ay, okay, may isa pa akong example ng mutation. Okay. I am lactose intolerant. Okay? Ayan. So, ano ibig sabihin ng lactose intolerant? Pag kumain ako or uminom ng anything with dairy, no? Ayan, with milk, no? In 30 minutes, tatakbo na ako ng CR, no? Ayan. In fact, kagabi, I ate this very delicious tuna with mushroom steak, no? Hindi ko alam na yung tuna with mushroom steak pala may halong cream, no? Eh, ang sarap. So, dinire-diretso ko yung kain. So, kaninang umaga, no, naka-multiple times ako doon sa CR. Ayan. So, ganun kalala yung lactose intolerance ko. But did I finish the food? Yes, no? Masarap eh. Ayan. Ang sarap ng tuna dito, no? Ayan, sa CDO. Ayan, I really enjoyed eating it a lot. Ayan. Anyway, I am lactose intolerant. And do you know that lactose intolerance is actually the wild type? Ayan. So, pag ikaw ay hindi lactose intolerant, ikaw ay mutant. Okay? Ayan. Bakit? No? Bakit? When you are young, no? When you are young, you get to tolerate milk. Kasi you need the milk, no? As a nutrition, as a complete nutrition. Okay? Kasi hindi ka pa kumakain ng solid food. Okay? Now, over time, humans, no, humans, I'm talking about humans, so humans would lose you would lose the gene that encodes for the lactase, no? The lactase, no, is the one that digests your lactose. Okay? Yan. Pag meron kang lactase, you're good. You're not lactose intolerant. Okay? Ay, sorry. Pag may tama. Pag may lactase ka, you're good, no? You're not lactose intolerant. Okay? Kaso yun nga, normally, no, humans lose the gene for the lactase enzyme. Okay? Bakit? Kasi as you grow older, no, you get to eat solid food. Hindi ka naman na umiinom ng gatas, no? Hindi ka na required uminom ng gatas. Okay? Ayan. Kaya hindi mo na kailangan yung lactase, no? You get to have your proteins from solid food, no? Ayan. So, other individuals, no, don't get to lose yung lactase nila. Okay? Yan. So, they are the mutants. Okay? So, yung mga mutants, no, sila yung meron pa rin napoproduce na lactase sa stomach nila. They are not lactose intolerant. Tapos yung mga wild type, yung mga normal, okay, kami yun. Okay? Ayan. So, yung mga normal, naglo-lose ng lactase, okay? So, dahil nag-lose ka ng, ang pangit na superpower, no? So, their superpower is not lactose intolerant, no? Ayan. Ayan, bilang mutant. So, pag lactose intolerant ka, you're what type, no? Kasi you don't have lactase. So, ang nangyayari is, yung lactose in your stomach will now be digested by bacteria. The bacteria will convert your lactose into lactulose, which will again be digested by the bacteria to form your acetic and lactic acid, no? Na siya yung nagkakos ng pagpupoops, no? Ayan. So, yun yung nangyayari, no? Kung bakit ka lactose intolerant. Okay? Yeah, so I'm proud to say that I am wild type, no? Ayan. So, when it comes to my being lactose intolerant, okay? So, ang pangit na superpower, no? I am lactose tolerant, no? Ayan. So, I get to drink milk. Ayan. Yun ang superpower mo. Kaya mo uminom ng gatas, no? Ayan, di lang isang mutant. Okay, so, may nag-chat pala. Okay, ano yung chat niya? Ay, wala. Okay. Okay, so now let's proceed. Ayan, nakwento tuloy ako. Okay, so meron tayong tinatawag na substitution, substitution, deletion, and insertion. No? So those are the three different types of mutation. Let's begin with substitution. So when you say substitution, a base, no, is replaced by another base. Okay? Ayan. So a nitrogenous base is replaced by another base, no, in your nucleic acid. Okay? That is your substitution. Now there are two types. You have transition and you have transversion. So when you say transition, no, purine is converted into another purine. Okay, let's say adenine is converted into guanine and vice versa. Okay? Or a pyrimidine is converted into another pyrimidine. So let's say cytosine is changed into thymine, no? Or thymine is changed into cytosine. All right? So ang tawag doon transition. Okay? And then pag sinabing transversion, okay? Ayan, transversion means a purine gets changed into a pyrimidine and the pyrimidine changes into a purine. Okay? So for example, if you change or if you deaminate your cytosine, no, the cytosine can be read as thymine. Okay? Yeah, so the cytosine can be read as thymine. So example noon is this one. No? So if this is your cytosine, no, in the presence of UV light or high temperature, the cytosine can actually be deaminated So matatanggal yung amine group dito. And it can be read as your thymine. Actually, it's uracil, but of course, in the DNA, it is read as a thymine. Okay? And the thymine will then combine with your adenine. Okay? So, for example, I have GC. Okay? The GC becomes AT. Okay? So remember that deamination of cytosine results in a GC to AT transition. Okay? So bakit naging GC to AT transition? Kasi yung cytosine, it is read as a thymine. Okay? Ayan. So your cytosine is read as a thymine which combines with your adenine. So ito yung cytosine. Ayan. Tapos mag-prepare yung thymine with adenine. Okay? Ayan, kaya instead na GC, yung nandoon sa gene, nagiging siyang AT. Okay? So that's the GC to AT transition due to the amination of cytosine. Another is the amination of adenine. Ayan, so this is your adenine. If we deaminate your adenine, it will produce your hypoxanthine. No? Now this is the enol form of your hypoxanthine. No? So meron pa siyang isa pang form. Yung isa pang form nito ay lumilipat lang yung double bonds. Ito lilipat dito, ito lilipat dito, ito lilipat dito. Thus this becomes, hindi na ito lilipat doon. Try natin. And then you have here a double bond. Magiging double bond po. Alright? Ayan. So pareho lang yon. Okay? So hypoxanthine can be read as G. Okay? And then the G will have a propensity to combine with cytosine. It also results in GC to AT transition. Okay? So bakit naging GC to AT transition? The amination of adenine, so yung adenine, okay, madi-deaminate siya. Okay? So pag na-deaminate yung adenine, it will bond with your cytosine. Okay? Ito yon. Okay? Kasi yung adenine mo ay napagkamalang guanine. Okay? Kaya ulitin natin. When you deaminate your adenine, it will produce hypoxanthine which can pair with cytosine. Okay? So yung cytosine ay magbabond eventually with guanine. Okay? Ayan. So it also occurs or it also results in GC to AT transition. Or baliktarin natin, AT to GC transition. Ayan, para mas malinaw. Ayan. Okay, so clear ba yun? Ayan, so those are transitions. Okay? Ayan, basta transversion, version, no? So, bagong version. Okay? Purine to pyrimidine. Okay? Now, what are the effects of base substitutions? No? So, you have missense, nonsense, and silent mutation. Okay? So, pag sinabi nating silent, nasabi ko na to kanina, pag silent mutation, napalitan yung base ng isa pang base, but the codon still encodes the same amino acid. Okay? That is a silent mutation. Another silent mutation is when the mutation occurs in the non-coding region. Okay? Remember that the mRNA is composed of your untranslated region at saka translated region. Kapag nasa untranslated region yung nag-mutate, di wala tayong problema. No? Hindi naman siya matatranslate. No? So it's a silent mutation. Okay? Now, you also have your nonsense mutation. So pag nonsense mutation, the mutation results in the formation of your UAA, UAG, and UGA. No? Ayan. So, stop. So you have a cut or you have truncated protein. No? Nonsense. No? Ayan. So kumbaga nonsense yung protein kasi it's not functional. Okay? Yan, nonsense. Then missense mutation means na it's different. Okay? Yan. So what happens here is that you change the amino acid product. And this happens in sickle cell anemia. Okay? In sickle cell anemia, we have the GAG, no? We have the GAG which encodes for glutamate. Okay? And then in sickle cell anemia, nagkakaroon tayo ng AT transition. Okay? Ay, sorry, hindi transition, mutation. No? Ng AP mutation. Anong klaseng mutation is ito? Is this transition or is this transversion? Yung AP. This is transversion, no? Kasi you have a purine that becomes a pyrimidine. Okay? Ayan. So you have here the transversion, no? So yung A naging T. Alright? Ayan. GAG and ayan. So, may audio na po ba? Saan parte na wala yung audio? Wait lang. Search tayo. Hello. Sana wala yung audio. Pagka-draw. Okay, wala naman ako masyadong nadagdag. Okay, so in sickle cell anemia, okay, so what happens is, ayan, there is one basic change, no? GAD to GPG, no? That's a transversion, no? From adenine, it becomes thymine. And yung glutamate, nagiging valine, no? So valine is a hydrophobic amino acid. So it results, no, in the clotting, no, or clogging of the hemoglobins, no, because of hydrophobic interactions. Okay? Yeah. So basically, yun lang na. All right? And so, next part. Ayan. So, these others, no, deletion and insertion. So, when you say deletion and insertion, medyo self-explanatory, no? Deletion, one or more nucleotides are removed from the sequence, or insertion, one or more nucleotides ay inserted in the sequence, no? Ayan. Normally, they occur at the same time, kaya we call them indels, no? Indels, insertion and deletions. Right? Now, insertions and deletions result in, may nakaharap pa na. So insertions and deletions result in your frame shift mutations. No? Yeah, so when you say frame shift mutations, nagbabago yung reading frame. Di ba na-mention natin yung kanina? Yung reading frame, no? Yun yung binabasa, no? Natin, no? To translate the mRNA, no? Now, if you remove, no? If you delete, no? A certain portion of it, pwede magbago yung reading frame. Okay? Dahil nagbago yung reading frame, pwedeng iba na yung protein sequence that will be produced, no? For example, ayan, gawin natin yung example. I have GA, G, T, A, C, PGA. No? Ayan. So, kunwari ito yung DNA. No? Ayan yung DNA. Kunwari na-delete, no? Kunwari ang na-delete ay dalawang bases. Okay? So, magbabago yung ating reading frame. Okay? So, ang magiging bagong reading frame ay GA, ayan, C, P, G, and then may A on. Okay? Ayan. So, magbabago yung yung photons, no? That will be produced, no? So, yan yung frame shift mutation. Now, normally, frame shift mutations also happens because of a faulty polymerase, no? Yeah. And this usually happens if there are repeated nucleotides in the sequence. Ito yung tinatawag natin na trinucleotide repeats. No? Ayan. So, pag maraming repeats, no, in the sequence, no? For example, pag meron kang CAG repeats, that's Huntington's disease, or yung CGG, that's your Fragile X syndrome. Now, pag mas mahaba pa, no, let's say pare-parehong nucleotide, C, 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 ang daming C. Okay? Ang tendency, no, the tendency of your polymerase is, i-assume niya na tama na yung dinaanan niya. No? Eh, parang ikaw din yan. No? Yan. Diba, kunwari, naglalakad ka. Tapos pare-pareho nakikita mo, uy, parang galing na ako dito, sige, talo na. Yan, no? Ganun yung mangyari, di ba? So, kunwari pare-pareho ba yung itsura, no? Ayan. Parang siya, diretso. Sige, talo na ako, no? Parang dito eh, di ba? Yan. Hindi mo na makalala. Ganun din yung DNA polymerase, no? So, actually, as much as possible, we have to avoid repeats. No? Ayan. So, dapat wala yung mga yan. Kasi tatayo yung DNA polymerase. Ayan. Now, mutagens are the agents that cause mutations. This can be, mutations can be caused by any of the following. We have oxidants. For example, mga oxidizing agents, no, can convert your guanine to oxoguanine, which results in a transversion mutation. All right? Ayan, so yung oxidation ng guanine, no, would make it appear like a thymine. Okay, yan. So, yung thymine will form a base pair with adenine. No? So, G2T. Okay? Guanine to thymine, so that's a transversion mutation. Now, nitrosamines and nitrous acid can also cause the amination of cytosine, adenine, and guanine. Okay? Yeah. Yung nitrous acid, this is a very common preservative in meat products. Okay? Yan. So, di ba kaya sinasabi nila, no, hinay-hinay daw yung pagkain ng tosino na, ano pa ba, tosino, tama, no, yung mga ganon. And anything that's preserved, ano ba ito nila? May chat ba ulit? Yeah. Hindi clear kasi wala na akong microphone, nag-charge siya. So, gaano kasama? Nag-message lang, hindi raw clear. Ay. Ay. Ay, sige, sige. Wala pa. Okay. Sige, message mo na lang. Hindi pa ako tapos eh. Ayan. Okay, so, ayan. Malapit na kayo mag-quiz. So, wait lang. Ayan. Kasi wala na akong microphone na nagagamit. Okay. Nag-charge pa siya. Okay. So, what I was saying is that yung nitrous acid, no? Yan, lalapit na ako sa microphone. Yung nitrous acid ay galing sa sodium nitrite and potassium nitrite. Ito yung inyong salitre. No? Ayan. So, they are common preservatives for meat products. Now, the meat products contain proteins. No? Proteins interact or react with the nitrous acid to form nitrosamines. Ito. Okay? Ayan. Nagets? So yung nitrosamines and nitrous acid, they can cause the amination, removal of the amines in your cytosine, adenine, and guanine. Another mutagen is your alkylating agent. No? So pag sinabing alkylating agent, ito yung mga nag-a-add ng methyl or any alkyl groups to the bases. No? For example, yung uracil, pag na-methylate yan, it will become thymine. Even cytosine, pag siya ay na-methylate, no, it will look like thymine. For example, a methylated guanine will look like adenine. Okay? That's a transition mutation. And then UV rays, no? Yung UV rays naman is it can cause thymine dimers. No? Ayan. So yun yung dapat yung tandaan doon, thymine dimers. Meaning to say, nagdidikit yung dalawang thymine, no, in a sequence. Okay? Forming a cyclobutane ring. Okay? And then finally, you have your ionizing radiations. Ionizing radiation ay yung X-ray and gamma rays. No? So X-rays and gamma rays are high energy radiation. Okay? They tend to break the nucleotides. Okay? They tend to break the bonds, no, in your nucleotides. Okay? So let me give you an example. Diba, lagi sinasabi sa MCU, sa Marvel Cinematic Universe, no? Si the Hulk daw na-expose sa gamma radiation. Yan. Tapos, dahil na-expose siya sa gamma radiation, hindi siya tahong. No? Yan. Do not ever try to expose yourself in gamma radiation. No? Kasi, you will die. Okay, yan. So, you will die kasi magbe-break yung nucleotides, hindi magre-replicate yung cell, mamamatay yung cell, deaths. Okay? Ayan. Ganun din si The Thing, yung Task Force. Ganun din daw yung nangyari. Mabas sila sa Earth, nasa tamaan sila ng cosmic rays. Cosmic rays are basically gamma rays. Okay? So, ganun din, no? Hindi totoo yung mga yun. Kaya panira sila ng mga childhood memories, no? Yung knowledge. Ayan, sana hindi na natin alam. Ayan. So, UV ray is a non-ionizing radiation. Okay? So, para lang clear, UV ray is a non-ionizing radiation. Okay, so last slide for this part. Then, pwede na tayo quiz. Ayan. Okay. So, mutation repair. So, meron different types of repair. Direct repair. No? Basically, you remove the wrong base, you replace it with a new base. Okay? And in bacteria, they would require photolysins, no, for this to occur. There is also the methyl-mediated mismatch repair. No? So, basically, for this one, okay, the daughter strand is unmethylated. No? So, methylated yung parent strand. Okay? So, ano yun? No? Ibig sabihin, no? I-identify natin yung methylated strand. Yun yung totoong template. Okay? The mutation, no, which occurs in the unmethylated daughter strand, okay, must be avoided. Okay? Dapat hindi na natin siya i-replicate pang muli. Alright? Ayan. So, yan yung purpose na methyl-mediated mismatch repair. No? So, para malaman ng body mo, sino yung totoo? Kasi di ba, remember, replication is semi-conservative. No? In one DNA strand, you have a parent strand and you have a daughter strand. Right? So, para malaman, no, ng DNA polymerase, sino yung parent strand, dapat methylated siya. Yung unmethylated, yun yung daughter strand. So pag may mutation, most likely it will be found in the unmethylated daughter strand. Okay? Yeah. So hindi niya gagamitin yung unmethylated daughter strand. Okay? Then we have the base excision and the nucleotide excision repair. Yan. Base excision repair changes. No? This requires DNA glycosylases. And yung inyong nucleotide excision repair naman utilizes exonucleases. Okay. Now, xeroderma pigmentoso, or XT is actually a condition wherein a very slight exposure to UV radiation would result in skin cancer. Okay, yeah. This is not very common, no? This is a very rare disease, no? In fact, among the documented cases nito, isa lang yung pinakasikat, no? One girl from China, okay? And she's known as the girl who lived in the dark, okay? Yun yung tawag sa kanya. Bakit? Kasi she actually lives up there in the mountains in China, no? Tapos, hindi alam ng parents niya kung ano yung balik sa kanya, okay? Pero tuwing lumalabas siya bahay, no, nasusunog yung balat niya. And not just sunog, no? Sunog na sunog. Okay? Ayan. Normally, we have this excision repair. Meron tayong nucleotide excision repair. So even if we are exposed to UV radiation, certain levels of it, we can tolerate. Okay? So certain levels of UV radiation, we can tolerate. Yung individuals without NER, no, hindi nila talaga kayang i-tolerate kahit anong level of UV radiation. Alright? Kaya nagkaroon siya ng skin cancer. So now, no, ang nangyari is, yung balat niya, parang sunog-sunog, nagbilog-bilog, no? There are, namatay siya because of bacterial infection. Okay? Kasi yung cell, yung tissues, yung skin niya, na hindi exposed to the air, nagkaroon ngayon na mga anaerobic bacteria, no? Ayan. So, yun yung kinamatay niya. No? Ayan. So, actually, nung na-document siya, no, naka-cover siya, no? Para siya hazard suit, no? Naka-hazmat siya, ganyan. Kasi nga, bawal siya expose sa light. Kasi yung bahay nila, nakapatay lahat ng ilaw. Kasi even fluorescent lang releases UV radiation. Okay? Ayan. Naapektuhan din siya. No? Ayan. So that is the importance of your nucleotide excision repair. Alright? Ayan. Okay, so that's it for this part. Ayan. Magkakaroon lang muna tayo ng short na review, no? Ayan. So, daanan lang natin, no? Para ma-ready kayo for the quiz. Ayan. So, sasagutin ko muna natin ito. Ayan. Alright. Teka lang, ha? Wait lang. Hindi yan yung, hindi yan yung para sa. Parang alas ganun din. Alright. And so let's session muna. Oh, that's okay, so next one. The fatty acids are considered essential fatty acids except A, BPA, B, EPA, C, alpha linolenic acid, or D, oleic acid. So what's your answer? A, C. Ayan. May narinig ka ba akong tamang sagot? The correct answer is letter B, oleic acid. Yeah, so remember, no, all of these are essential fatty acids except your oleic acid, right? Remember, essential fatty acids are fatty acids that are derived from the diet. Right? Yeah, pag napoproduce ng body, no, yan ay hindi essential. So yung mga essential amino acids ay sina linolenic acid, linoleic acid, BPA, and DHA. Alright? Yan. Okay, number 2. Which is the correct abbreviation for gamma linolenic acid? A, 16-1-9C. B, 18-2-9C-12C. Letter C, 18-3-6C-9C-12C. Or D, 18-3-9C-12C-15C. Letter C. The correct answer is letter C. Ayan. So remember, sinabi ko kanina ay kahapon, linolenic acid na naman yan. The alpha and the beta. Yung alpha, 9 yung start. Okay? Tapos si gamma, 6 yung start. Okay? Tapos plus 3, plus 3. Alright? Yan. So C is our correct answer. Okay. Next, number 3. So number 3, which are the primary lipids present in very low density lipoproteins. Ayan. So very low density lipoproteins would have your actually, hindi ko pala natin na-discuss ko, pero sige. Ano? This is PFAS. This is letter C. Kaya ayaw ko naman. Letter C. Triglycerides. Okay? Ayan, so, very low lipoproteins. Okay? So, i-discuss pa naman natin yan. Don't worry. Okay. Next number. Number 4. Which are found in cell membranes and contain two fatty acid moieties esterified to a glycerol backbone. A. Cerebrosides. B. Gangliosides. C. Phosphoglycerides. D. Sphingolipids. Found in cell membranes. Yeah, found in cell membranes yung key. Two fatty acid moieties. Ayan, attached to the glycerol backbone. Ayan, your answer is letter C, no? And C, phosphoglycerides. Remember, phospholipid cell membrane, no? Sabi ko yung kahapon, no, you have the lipid bilayer, which is consists of the phospholipid. No? Sabi ko, hindi siya sperm. No? Yan. May dalawang fatty acyl, no? Na tail. Ay, shucks. Alright, so, number 5, the combination of sphingosine and fatty acid is that is okay. Yeah. So, sino si cardiolipine? Cardiolipine is diphosphatidylglycerol. Okay? Tapos si cephalin, sino si cephalin? This is your phosphatidyl ethanolamine. No? Ayan, phosphatidyl ethanolamine. Your choline is it's the alcohol. It's an amino alcohol. Okay? Ayan. So, choline. Ayan. Number 6. Blank is characterized by a deficiency of lysosomal hexosaminidase A, resulting in the accumulation of gangliosides in the brain. Ayan. So, pag gangliosides in the brain, ano ba yung gangliosides? Ito yung may sialic acid, di ba? Sialic acid, ganglioside. Ganglioside. Letter D, no? Sialic acid. Alright. Okay. So remember yung Gaucher's disease, ano yung kulang sa kanya? Yung glucocerebrosidase. Beta glucocerebrosidase deficiency. Then yung Crab's disease naman, beta galactocerebrosidase. Okay? Pag sinabi natin cerebro, no? Ibig sabihin nun, yan ay monohexoceramide. Okay? Niemann-Pick disease is the accumulation of your phospholipids. Okay? Ayan. Number 7. Hindi naman siya gumana. Okay, so pagdating natin dito. Ayan. Which is the most widely occurring sterol? A. Cholesterol. B. Ergosterol. B. C. Beta-syntosterol. Or D. Stigmasterol. A is correct. Ayan. A. Ayan. So, B. Ergosterol. Fungi, no ergo, no? Although it can also be found in plants. Ano pa yung alam natin kay ergosterol? It is the provitamin D2. Cholecalciferol is your provitamin D3. Okay? Then beta-stigsterol, most important sterol in plants. Okay, stigmasterol, present also in plants. Number 8, which is the primary precursor of fatty acids? Ulit-ulit ko sinabi ito kahapon. Letter A, acetoacetate. B, acetyl-CoA. C, isopentenyl diphosphate, or letter D, pyruvate. The answer is letter. Anong pathway na gagawa ang fatty acids? Sa acetate pathway, and the precursor of your fatty acids ay acetyl-CoA, diba? Ayan, remember, that's the reason why carbons are always added, no, to the chain of your fatty acid. Kasi acetyl-CoA yung kanyang precursor. Alright? Ayan, number 9. which is synthesized from arachidonic acid via the enzyme 5-lipoxygenase. Ayan. Sino dyan? A, bile acids, B, leukotrienes, C, prostaglandins, or D, steroids. So, bile acids and steroids are both steroids. Okay? Bile acid is a steroid. Ayan. So, yung steroids ay napoproduce from what? Precursor, your methylonic acid, diba? Yan. So sabi natin, 3 molecules of acetyl-CoA will form your methylonic acid, which will be converted to isopentenyl diphosphate and dimethyl allyl diphosphate. No? And then that will eventually produce the cyclopentanoperhydrophenanthrinate, which will produce your steroids, including bile acids. Okay? So, hindi yan. Mali na yung A at yung D. So, B and C na lang. Arachidonic acid via the enzyme lipoxygenase. The answer is letter, mali pa rin, letter D. Leukotrienes. Remember, prostaglandins are produced via cyclooxygenase pathway. Okay? Well, so, arachidonic acid plus arachidonic acid in the cyclooxygenase pathway will produce your prostaglandins. Okay? Now, kapag leukotrienes, sino yung immediate precursor ng leukotrienes? Your 5-hydroperoxy ecosa tetraenoic acid. No? Ang haba no? Ayan, susulat natin. So remember na for leukotrienes, for leukotrienes, ang immediate precursor niya, for leukotrienes, yung immediate precursor is, ayan, 5-HPEPE, no? Ayan. So, arachidonic acid, tapos, lipoxygenase enzyme, okay? So, 5-HPEPE. And this is your 5-hydroperoxy. Peroxy, ecosa, tetraenoic acid. Alright? Ayan. So, yan yung H-hydroperoxy, ecosa, tetraenoic acid. Via the lipoxygenase pathway, leukotrienes. Alright? Okay. Next number. Number 10. Ayan. If the base sequence of a segment of DNA is CAG, TTA, GC, which of the following sequences is complementary? Try nyo muna. Ay, ano na? What's your answer? Sino nagsasabing A? Ba't bago nyo? Teka, bakit wala nag-class, wala nag-class? Sino nagsasabi letter B? Wala rin. Sino letter C? Letter D? Wala sagot. Alright. So, sige, sagutan natin. This is 5' to 3'. So, CAG, TTA, GE. Okay? This is 5'. This is 3'. So, ngayon, susulat tayo na pabaliktad. Okay? So, this is 5'. This is 3'. G, C, T, A, C, E, G. Pag ganito yung basa natin, sino siya dyan? G, C, T, A, C, T, G. That is letter A. Okay? So letter A is our correct answer. Na gets? Gets na? Okay? Ayan. So last 5 numbers. The structure shown below is which of the following compounds? A, adenosine. Letter B, oxy, guanine. Letter C, thymine, delete. Letter D, cytidine. Or letter E, uridine. Sige, I'll teach you how to analyze. Right? Ano-ano yung components in the structure? You have the base. May base tayo. You have the sugar. And then you have the phosphate. So, sino to? Is this a nucleoside or is this a nucleotide? This is a nucleotide. Okay? If you have a nucleotide, what should be the suffix? Pag-purine. Pag-purines, ethane. Diba? Pag-pyrimidines, ethylate. Ayan. So, hanapin na natin kung sino-sino sila. Ay, pwede ito, ethane, ethane, ethane. Eliminated na to. Dali, diba? Next. Okay? Look at the sugar. Is the sugar 2-deoxyribose or is it ribose? Is this 2-deoxyribose or ribose? This is position 1, this is position 2. May OH o wala? Meron. so, ulit. Is this ribose or deoxyribose? This is ribose. No? Kasi may OH sa 2 prime. Okay? So, ribose. So, papagkanin na natin yung deoxy. Mali na yan. Okay? Diba? Mali na. Tapos, letter C. C. Mali din. Ang thymine laging nasa DNA. Deoxy siya palagi. So, C is also eliminated. So, letter E. Diba? Yan. So, even though hindi natin talaga alam yung structure, okay? The fact na na-analyze natin siya, you can already eliminate, no? Ganun yung gawin nyo, okay? Now, sige, tingnan natin yung base. No? If you look at the base, ayan, di ba sabi ko pag nakadapa, ganon, dalawa lang yun eh. Dalawa lang yun. Uracil and thymine. Di ba? Dalawa lang. Pero yung thymine has a methyl group over here. Okay? Yeah, so then wala siyang methyl group, that is your uracil. So, letter E, uridine. Okay? Nagets? Ayan. Okay. So, number 12. Which one of the following is true of double helical DNA? Ayan. Letter A. The planes of the bases lie parallel to the helical axis. Ayan. So, true or false? The planes of the bases lie parallel to the helical axis. Ayan. This one is the planes of the bases lie parallel. Hindi, no? The planes of the bases lie perpendicular to the helical axis. Diba? Yan. So remember yung drawing natin kahapon, pag ganito, the bases are like this. Okay? So this one is false. B. Okay, letter B. The chains have a backbone of linked glycosides. Linked glycosides, ang backbone natin ay sugar, phosphate. Diba? Phosphodiester bonds are in the backbone, so B is not correct. Okay? Letter C. Unless the DNA is circular, the 3' hydroxyl groups of each chain are at opposite ends of the structure. Anong tawag natin dito? This is the anti-parallel, no? This is the anti-parallel arrangement of the 2 strands of your DNA. So this one appears to be correct. Okay? Pero hindi pa natin sure. Tingnan natin kung may iba pa. Letter D. The duplex structure is stabilized only by hydrogen bonding between bases. While it is true that the most important factor that stabilizes the duplex DNA hydrogen bonding, hindi lang yun. Also, other van der Waals interaction come to play. So letter D is not correct. Ayan. Letter E. At saka clue pala na pag nagtitig kayo ng exams, pag nakakita kayo ng only, yung word na only, okay, kadalasan mali yan. Kasi, ibig sabihin, meron pang iba na tama. Ayan. So, yan. Clue yan. Letter E. Although they are associated in anti-parallel fashion, the two chains have identical base sequence. Identical ba yung dalawang shades? No, no? Hindi naman madalas. So letter E is wrong. So only C is the correct answer. Alright? Ayan. Okay, next. Number 13. The melting temperature of DNA is best defined as being letter A proportional to the length of the DNA. Letter B, proportional to the guanine cytosine content of DNA. Letter C, the temperature at which all of the DNA is denatured. Letter B, proportional to the purine content of DNA. Or letter E, the same in all animals. So letter E is wrong. Alam natin yan. Okay? Kasi magkaiba yung sequences. No? So magkaiba rin yung melting temperature. Alright? Proportional to the purine content. Sino ba yung purine? That's adenine and guanine. Di ba sabi natin, what dictates the melting temperature ay guanine, cytosine content. Okay? So, letter D is wrong. Now, letter C, the temperature at which all of the DNA is denatured. Okay? Is this true? Identify natin ito kahapon. Diba? Denaturation is the temperature at which 50% of the DNA is single-stranded. So letter C is wrong. Aha, tanong na lang. Okay, ayan. So between the two, okay, sino yung mas tama dito? Yung A, tama naman siya, no? It's proportional to the length of the DNA. However, it's not the most important factor. Okay? What if mayroon kang dalawang chains, yung isa puro A and B? Pareho siya ng size or length ng isa pa DNA na puro G and C. Sino yung mas mahaba? Sino mas mataas ang melting temperature? Same lang sila ng length, ha? Yung puro AT or puro GC? Yung puro GC, no? So A is not correct, no? Ayan, kasi what defines the melting temperature of DNA is actually the guanine cytosine bond. Alright? Although, the length also comes into play, no? Ayan. Basta pareho lang sila na composition. Okay? Number 14. If the cytosine content of a double helical DNA is 20% of the total bases, the adenine content would be ayan, 10%, 20%, 30%, 40%, or 50%. Eh, isipin ninyo, sino yung nagsabi, no? Or ano yung sinabi ni Erwin Schargat? According to Erwin Schargat, no? There is 1:1 ratio between adenine and thymine, as well as guanine and cytosine. So, if the cytosine content is equal to 20%, what will be the percentage of guanine in the DNA? 20 rin. Okay? So, kung 20 plus 20 is equal to 40, ilan na lang natira? 60%. No? Now, the 60% will be divided by how many bases? 2. No? Adenine and thymine. Dalawa na lang. So, we divide this by 2, so you get 30%. So, the answer is letter C. Ayan. Okay? Last number, yeah. If a sample of DNA is found to have the base composition of adenine equals 40, thymine equals 22, guanine equals 19, and cytosine equals 19, what conclusion can be drawn? Letter A, the DNA is circular duplex. Letter B, the DNA is linear duplex. Letter C, the DNA is single-stranded. Letter D, the DNA has highly repetitive sequences. Or letter E, the DNA has a high melting point. Ayan. Now, if you look at the percentages, hindi equal yung adenine at saka yung thymine. No? Hindi rin equal yung guanine at saka yung cytosine. Posible bang duplex DNA ito? No, no? Kasi pag duplex DNA, sabi ni Richard Duff, dapat equal. Okay? So lahat ng may lahat ng may duplex hindi. Duplex, hindi. Okay? Yeah, so X. Okay? Yes? Ayan, next. Tingnan natin letter D. The DNA has highly repetitive sequences. Kung highly repetitive yan, hindi dapat meron dyan sobrang taas, no? Ng percentage. Pero wala. No? So this is also wrong. Ayan. The DNA has high melting point. Posible ba yun? Based on the GC content, can we say that this has a high melting point? No, kasi 19 UGC, no? Mababa, no? So wrong dito. So the answer is letter C. The DNA is single stranded. Alright, ready na? Ayan. Okay, so to give you time, sige, let's have a 10-minute break. Pagbalik, no, yun yung quiz. Okay? Ayan. So kayo bahala how you would want to use your 10-minute break. Okay? Ayan, kung gusto niyo bang mag-review or gusto niyo mag-break na muna. Okay? Ayan. So we'll have a 10-minute break. Mag-search din ako kasi yun. Kailangan ko yung Ayan. Ano yun? Teka lang, ha? Alright. Okay, so let me go back doon sa sinasabi ko. Ayan. So glutamate, no? Glutamate comes from wheat. Kasi your glutamate, no, ay galing sa gluten. Ayan, gluten, ayan, which is the protein that is abundant in the wheat, no? And in fact, gluten is the one responsible for the glutinous, no? Yung malagkit na property of certain grains, no? For example, yung oatmeal, di ba, lumalagkit siya, yan, and yung wheat, no, it's because of the gluten. Now, some individuals cannot metabolize gluten properly and they cause inflammatory reactions to gluten, okay? Or they have inflammatory reactions to gluten. And those people ay yung merong sakit na tinatawag nating celiac disease. Ayan. So they have inflammatory reaction against gluten. That's why a common health food nowadays ay yung tinatawag nating gluten-free, no? Yan, kasi may mga individuals na nagkakaroon ng inflammatory bowel, no, because of the gluten in wheat. Now, aside from these, no, you also have your glycine, no? So glycine is sweet, no? Because it's the smallest amino acid, and glycos, no, means sweet, no? Ayan, so glycine, no? Galing doon sa term na yon. Now the last amino acid to be discovered was threonine, which is an alcohol amino acid, and asparagine is the first to be discovered, ayan, which I will show to you yung structure niya. Ayan. So let's look at their structural features. Now, all amino acids have the same structure. No? They are all alpha amino acids. Okay? So when we say alpha amino acids, they have alkyl groups, or they have functional groups at the alpha position. Now, remember that the alpha position is the carbon beside the functional group. Okay? Ayan. So the alpha carbon is the carbon beside the functional group. All right? So yung alpha amino acids, no, they have the carboxylic acid functional group and of course the amino group, both of which are attached in the alpha carbon of the molecule. The R group, which is the rest of this molecule, ay yung R group na that makes amino acids distinct from one another. Okay? The physical-chemical properties of amino acids are dependent on the structure of the R group. No? Ayan. So for example, the size of the amino acid, the charge or the polarity or even the solubility of the amino acids depend on the R group. Ayan. Now, let's begin with, ayan, parang ito lang matatapos muna natin, no? Hanggang sa pagkilala sa kanila. Let's begin with your non-polar aliphatic or group containing amino acids, okay? So remember that we can classify amino acids into this, no? You have the hydrophobic and you have the hydrophilic amino acids, all right? Yeah, so among the hydrophobic amino acids are amino acids, no, that contain only, no, non-polar aliphatic groups and those that contain the aromatic ring. Yeah, or the aromatic amino acids. All right? Now, among the hydrophilic amino acids, teka lang, 12 minutes pa. Ayan. So, among the hydrophilic amino acids are your amino acids that contain polar groups. And, for example, those that contain the hydroxyl group, OH, the thiol group, SH, no? And also those that are charged. Okay? So, meron tayong polar uncharged amino acids and the polar charged amino acids. The charged amino acids are your acids or acidic amino acid, and then meron din tayong basic amino acid. All right? Ayan. So we can classify your amino acids into these. All right? Now let's begin with your non-polar, no, aliphatic R groups or amino acids containing the aliphatic R groups. Okay? So you have here the following amino acids, no? Now the smallest amino acid which do not have the R group, okay, wala siyang alkyl group in the alpha carbon, it's your glycine. Ayan. So yung naka-shade na portion, siya yung R group, no? The only amino acid without an alkyl group in the alpha carbon is glycine. And because of that, it is also the smallest amino acid. Okay? And because your glycine is the smallest amino acid, it is also polar. No? Ayan. Actually, it's quite polar. Now, you can see at the bottom, ito, you have here numbers. Okay? These numbers are the hydropathy index numbers. Hydropathy index. So, ang ibig sabihin nito, the higher the value of the hydropathy index, the more hydrophilic, the more hydrophobic or lipophilic is the amino acid. Alright? Ayan. So, you can see na yung glycine, negative yung kanyang hydropathy index. It means it is quite water soluble. No? And it's because of its very small size. Okay? Now, aside from that very special feature of your glycine is also the only amino acid that is achiral. Ayan. Pag-uusapan natin ito mamaya, no? Chirality. Ayan. So, tandaan nyo na lang muna, glycine is the only amino acid that is achiral. It is not chiral. So, dahil hindi siya chiral, no? It does not rotate plane polarized light. It is optically inactive. Okay? Yeah, so your glycine is optically inactive. There's another property of glycine that is very important. Dahil it's very small and it doesn't have the R group and it is achiral, it can freely rotate. No? It can freely rotate on the alpha carbon. Pwede siyang mag-ikot-ikot ng ganyan. No? So you can find glycine at the turns of the coils. No? Yan. When you see a protein structure, pag nakakita kayo ng nagko-coil na protein, those terms, you will most likely find glycine, no? Yan, kasi nga, it is achiral. Alright? Yan. Now, let us begin, no, with alanine. Ayan. So, para madaling tandaan for me, I always count the carbon. Okay? Yan. So, ganun gawin nyo, no? Kasi again, do not memorize the structure. Okay? Be familiar with the structure. Okay? Ayan. So, simulan natin kay Alan, no? Ayan, may kapatid kasi si Alan, pero mamaya pa natin siya makikilala, no? Ayan, so alanine, no? It's the amino acid containing only methyl group at the alpha position. Alanine, okay? Ayan, one carbon. Okay, alanine, no? Isa lang 'yan. Okay? Now, punta tayo ngayon doon sa katabi niya, no? Yung katabi niya, this is your methionine. Okay? Now, in methionine, there are 3 carbons. Okay? And, ayan, your methionine is also the sulfide amino acid. Okay? It's the sulfide amino acid. So, pag sinabi nating sulfide, it is RSR. Okay? Ayan, so, R group, tapos may sulfur, tapos another R group. Okay? That's a sulfide. Okay? Kasi pag sinabi natin disulfide, no? Ito yon, R, S, S, R. This is your disulfide. Alright? Ayan. Methionine is the only sulfide amino acid in the 20 amino acids comprising your proteins. Okay? 3 carbons. Okay. Now, let's proceed to these 3. Itong tatlo, lagi magkakasama to. Leucine, valine, and isoleucine are your branched chain amino acids or the BCAAs. And BCAAs. No? These are your branched chain amino acids. No? Let's begin with valine. Okay? Ayan, si valine muna. Bakit si valine? Kasi 3 carbons din yan. Okay? 3 carbons. Okay? Yeah, so 3 carbons, tapos it forms a letter V. Oh, di ba? Ganda. Ayan, so valine, no? Letter V. Ayan, so valine. 3 carbons. Okay? Next, puntahan natin si leucine at saka si isoleucine. Okay? For leucine and isoleucine, they both have 4 carbons in the R position, in the R group, at the alpha position. 1, 2, 3, 4. 1, 2, 3, 4. Okay? So leucine and isoleucine are called structural isomers. Okay? So they are structural isomers. They have the same chemical formula, but the position of the methyl group is different. Okay? In both molecules. They are structural isomers. Alright? Now, ganito yung clue. No? If you draw the amino acid structure, yan, magbilang ka. From the carboxyl carbon, 1, 2, 3, 4, and 5. Okay? What is the middle of 5? 3. So, nasa 3 yung methyl group. Okay? That is your isoleucine. Okay? Kasi iso means balance. Gitna. Nasa gitna yung methyl group. Okay? Ayan, na-gets? Yan. So, sa pagbilang natin this time, kasali yung alpha carbon at saka yung carboxy carbon. Okay? So, at position number 3, you have the methyl group. Okay? That is your isoleucine. Balance. Nasa gitna. Alright? Now, yung kabilikan niya, no? Pag ito nilipat mo dito sa kabila, okay? That now becomes your leucine. Okay? Ayan. So, yun na yun, no? So, they are structural isomers. Okay? So, again, valine, 3 carbons, letter P. Tapos you have your isoleucine, methyl group at carbon number 3. Okay? And then, i-move mo lang yung carbon to 1 carbon to the next. Okay? You have your leucine. Alright? Pero ganito yung iniisip ng iba. No? Ayan. Yung ibang iniisip ganito lang. Pag leucine, letter Y. Okay? Ayan. Tapos yung hindi mo maintindihan yung structure, yun na yung isoleucine. Okay? Ayan. So, kasi ito, pag ganito yung isoleucine. Alright? Ayan. Okay? So, ganun, no? Para madaling ma-memorize yung structure. Alright? Now, may isa pa tayong hindi na di-discuss dito, and that is your proline. Now, proline is the only amino acid. Amino acid. Okay? So, anong ibig sabihin natin ng amino acid? Instead of having a primary amine at the alpha carbon, your proline has a secondary amine. Ayan, so meron siyang secondary amine. Ayan, ano bang ibig sabihin natin ng secondary amine? Ayan, so remember, kapag R and H2, ayan, this is your primary amine. Okay, R and H2 primary amine. Replace a hydrogen with an R group. No? So, ganito na yung itsura niya. R, N, H, tapos R. This is your secondary amine. There are 2 groups attached to your nitrogen. Okay, that's a secondary amine. So tanggalin mo yung isa pang H, that's already a tertiary amine. Okay? Now let's look at the structure of your proline. Yung proline, ito yung nitrogen. Meron siyang group dito, meron siyang group dito. It's a secondary amine. No? And bukod sa pagiging amino acid, secondary amine, your proline is also cyclic. No? Ayan. It is cyclic. Ayan. Remember, iba yung cyclic sa aromatic, ha? Ayan, magkaiba yun. Okay? So it is cyclic, pabilog siya. And because of that structure, you will most likely find proline at the bends of the proteins. So for example, meron akong protein, di ba? Kunwari yung protein ko, paikot-ikot siyang ganyan. You will most likely find at this portion, glycine, kaya siya nagko-coil. Tapos pag biglang nag-twist yung protein mo, malamang dito sa area na to, you have proline, kaya siya nag-bend. Okay? So kunwari ito nag-bend. Ayan, malamang dyan may proline. Okay? Ayan, so you can find proline at the bends or kinks. Kink, no? Sa kink, kulot. Sa kink of your proteins, no? That's your proline. Alright? So madali na bang ma-memorize yung mga amino acids with aliphatic R groups? Magsisimula tayo kay si glycine, etchapuera yun, no? Walang R group, no? Ayan, glycine, okay? Then we proceed to alanine, 1 carbon, okay? Tapos 3 carbons with sulfide group, that's your methionine, okay? 3 carbons, no, na aliphatic lang, that's valine, no, branched. Tapos 4 carbons branched, you have isoleucine and leucine. And then 3 carbons ulit pero cyclic, that's proline. All right? Ayan. Now, what are these letters, no, at the bottom of this slide? Ayan. So yung para sa mga non-polar aliphatic R groups, no, or for the amino acids containing non-polar aliphatic R groups, the 3-letter symbols, no, which denote the amino acid, ay yung 3 letters, first 3 letters of the amino acid. Okay? For example, glycine, gly, alanine, ala, proline, pro, methionine, meth, no? Leucine, leu, valine, val, isoleucine, dito lang nag-iba. Okay? Pangit kasing tingnan yung ISO, no? Ayan. Ang pangit no, ISO, no? Ayan, ISO lang. So ang ginawa nila ILE to mean it's isoleucine, isomer of leucine, ILE. All right? So again, para ma-generalize natin, para hindi kayo nagme-memorize, okay? For amino acids with aliphatic R groups, okay? The first 3 letters, no, would be their 3-letter designation, except for isoleucine, ILE. All right? Another thing, yung single letters naman. For the single letters, the first letter is their single letter notation. Yun na yun. Okay? So glycine is G, proline is P, methionine is M, valine is V, isoleucine is I, no? Leucine is L. Okay? 'Di ba? Madali nang i-memorize, no? Ayan. So, diyan muna, no? Natin. We'll continue after lunch. Ayan. Ayan, kasi mabibitin lang tayo, no? Pag dumiretso ako. Ayan. So far, any question? Ayan. Okay, sige. We'll proceed sa susunod. Maya-maya. Okay. Okay, I will pause the recording. Ayan, tapos